Today we're going to be looking at the cell membrane. And the cell membrane defines the boundary of the cell and determines the kind of contact that the cell has with its environment. Remember that the cell membrane is dynamic and flexible. We have this fluid mosaic model. It's made up mostly of phospholipid molecules, two layers, so we call it the phospholipid bilayer. And the way that phospholipids look is they have a hydrophilic head and a hydrophobic tail. And they arrange themselves so that the tails are in the middle and the hydrophilic heads are on the outside or on the surface. So you're going to have the hydrophilic or water-loving areas towards the outside and inside of the cell where we have very water-filled fluids or environments. Also within this phospholipid bilayer are embedded proteins. These are integral proteins. They could be channel proteins, carrier proteins, but they also may serve as receptors or enzymes or just a structural attachment for fibers of the cytoskeleton. They also could be attached to carbohydrates. There are plenty of carbohydrates found on the surface of cells, and they act like recognition molecules, telling your immune system that these cells are supposed to be in your body, that they're not foreign, that they're not trying to cause disease. So they're really good for surface reception as well as recognition. So when we're looking at the plasma membrane, we have to acknowledge there's an inside and an outside. If we're talking about stuff inside the cell, it is intracellular. If we're talking about stuff outside the cell, it's extracellular. So an intracellular fluid would be inside the cell, something like cytosol of the cytoplasm, whereas an extracellular fluid would be something like blood plasma or interstitial fluid. And all fluids, whether inside or outside of a cell, play a dynamic role in cellular activities. They help control what enters and leaves the cell. And the plasma membrane itself, also known as the cell membrane, is selectively permeable. That means it gets to decide what comes in and what leaves depending on the types of proteins, the types of carriers and channels that are within it. Really small things can go straight through the membrane, but larger things are going to need help. So here is that fluid mosaic model of the plasma membrane. You can see that we have a lot of phospholipids. So the red or orange circles, those are phospholipids. Their hydrophobic tails that don't like water are towards the middle. And then we see the hydrophilic heads in orange. They're facing away. We have different types of glycoproteins and lipids. So we have surface receptors, carbohydrate-based, that are on the outside or extracellular area of the cell. And notice that we have some cytoskeletal elements that are within the cell. So let's also look at the structure of the plasma membrane in just a little bit more detail. We have phospholipids. They make up about 75% of the cell membrane. And that gives us our lipid bilayer. The floating integral proteins are also known as membrane proteins. And those glycoproteins and glycolipids that act like surface receptors or recognition molecules in the extracellular space, those form a glycocalyx. The glycocalyx is just a surface sugar layer, kind of a sugar coat for cells that essentially you could see a little bit of under a light microscope, but it helps identify a cell. We just kind of give it this collective name, the glycocalyx. There's also going to be membrane structures that go within or traverse the plasma membrane that help the cell hold on to other cells. These might be extracellular matrix fibers. It might be parts of cell junctions, like desmosomes and their cadherin proteins. But overall, we do know that the fluid mosaic model is constantly changing. It's very dynamic. Not only do we have that double layer of phospholipids or a bilayer, it's also exceedingly thin. So it's a very specialized, fragile structure, and it really does rely on everything inside and outside of it, as well as embedded, like the protein molecules that are plugged into it, to make sure it works like it's supposed to. That not only is the plasma membrane a great barrier, not only is it protective, but it's also selectively permeable and helps the cell traverse its environment. So that lipid bilayer with the protein molecules dispersed in it, that's the fluid mosaic model. Things we have seen thus far, phospholipids, that's what it's made up of mostly. Cholesterol, I haven't mentioned that yet, but this is a steroid molecule, and it can play a role in how fluid a membrane is. And glycolipids are a derivation of the glycoproteins that we find on the surface of the cell. So great recognition molecules. And the glycolipids and the glycoproteins help make up that glycocalyx. And then here also is that information again on the phospholipids. Their head is hydrophilic, meaning it's polar. Something water-loving is polar like water. And their tails are nonpolar, like lipids, or hydrophobic. So that's why we see the tails sequester or hide away on the inside of the phospholipid bilayer, whereas the heads can interact with the watery interstitial fluid outside of cells and the cytosol, which is also watery inside of cells. Here's the basic phospholipid structure. Again, note that we have extracellular space up top, lumen or inside of the cell at the bottom, and that the heads, the hydrophilic heads, are facing both the outside and inside areas of the cell, and the tails are hiding away within the phospholipid bilayer. So again, 75% of the plasma membrane is phospholipids. The rest are glycolipids, so those surface molecules that are really good recognition proteins. And 20% of the plasma membrane is going to be cholesterol. And again, that plays a role in membrane stability by increasing or decreasing fluidity. This video is also going to have the PowerPoint posted. Make sure you check out Teams, and you can watch this video when you check out this PowerPoint there. So what are some of the other functions of the plasma membrane? We know it's a good barrier. We know it's selectively permeable. Well, it also helps control the electrochemical gradient. It helps maintain certain ions inside and outside of the plasma membrane, and it does so within a certain amount. And these ions have charge because ions are charged atoms, and the charge across a membrane can create electricity. That membrane potential can help create what's called an action potential, which muscle cells and neuron cells use. So more eventually on that electrochemical gradient, but it's one of the functions of the plasma membrane, its creation. We also know that plasma membranes are great communicating structures. Not only are there recognition signals that sit in the plasma membrane, cells can also hold on to each other or communicate via gap junctions that are embedded in their plasma membranes. And cell signaling is also possible via some of the integral proteins. So integral proteins include transport proteins, enzymatic proteins, or even receptors. A lot of these proteins are either channels or carriers, or some will bind to one molecule and then will initiate a process inside the cell without actually having that first molecule have to go through the cell. A lot of these integral proteins have areas that are hydrophobic and hydrophilic, so they can fit very snugly in the plasma membrane. There's also peripheral proteins. These are usually going to be focusing on whether or not the cell is holding on to something, so they definitely function in intercellular joining, cell-to-cell recognition, and also helping both cytoskeletal elements and extracellular matrix parts hold or attach themselves to different parts of the cell or things that are nearby. Here's another video that you could watch that will be in the PowerPoint that's posted on Teams. Another video, and there's several, so make sure to check out these videos that are posted on Teams that talk about all those integral proteins. So there's plenty of different cell structures that we've talked about. Now we've talked in more detail about the plasma membrane, so make sure when you're reviewing to come back to a table like this that goes through all the functions and different kind of distributions or locations for those cell structures. And I also highly recommend this video. It's by the YouTube channel called Amoeba Sisters, and it's an introduction to cells, so it's a great review for part 1 of this video lecture. So if you've forgotten some of what the organelles do or different types of cell junctions, make sure to go back and review part 1, and then also check out this PowerPoint in its entirety on Teams. But our next and main topic for part 2 of this PowerPoint is membrane transport. And that's when we talked more about the plasma membrane. We talked about the integral proteins, things like carriers and channels that are going to help move stuff across the membrane. There are 2 ways substances move across a plasma membrane, either via a passive process or an active process. A passive process does not require energy, whereas an active process does. An active process uses ATP energy. A passive process with no energy required is always going to move molecules from an area of high concentration to an area of low concentration, whereas active processes do the opposite. They move substances from an area of low concentration to an area of high concentration. So here is an image showing diffusion. Diffusion is when a substance without any energy added goes from an area of high to low concentration. The drop of dye initially added is not stirred, shaken, or anything like that. We're not adding any extra energy into these beakers. But that drop of dye will move from the area where it first enters the water, where it's highly concentrated, out towards the rest of the glass that's filled with water, because the rest of the glass is going to be an area where there is less concentration of that dye compared to the location where it initially is added to the cups. So more on passive transport. Passive transport relies on the fact that the plasma membrane is selectively permeable, that some things can get in and some things can get out, but not everything. This allows the cell to basically sustain itself. A cell that doesn't have selectivity wouldn't be able to make sure that it got certain food products inside or got rid of waste products. So passive transport that's naturally occurring, that requires no energy, consists of a couple of different types. There's simple diffusion, there's also going to be facilitated diffusion, and we'll look at these passive processes, as well as a passive process that moves water instead of solute. But all passive processes do not require energy. We're going to see that in facilitated diffusion, we're going to use proteins that act as channels or pumps, and they're going to pump something called a solute, which is the thing dissolved in a smaller amount, or something dissolved in a solvent. They're going to pump solute from high to low concentration. So looking more at passive transport, keep in mind again that plasma membranes have hydrophilic and hydrophobic regions, so that's why sometimes we're going to need help to move substances. But any substance that is lipid or fat soluble, or something really small, like maybe 1 molecule of oxygen or carbon dioxide, or 1 molecule of H2O. Those things are going to have an easier time just getting straight through the membrane by themselves without the help of a channel or carrier protein. Whereas something that is highly polar, something that is large or charged, like simple sugars, amino acids, ions, a lot of water molecules, they're going to need help. They're going to need a channel or carrier protein to help them move from high to low concentration across the plasma membrane. So we have a couple of different types of passive transport now to point out. Diffusion can be simple or facilitated. Diffusion is always the random movement of molecules due to kinetic energy, and that will push them from an area of high concentration to an area of low concentration, and they're going to be moving through a membrane. Simple diffusion does not need any carrier or channel proteins in order for it to happen. Something that is lipid soluble, something that is small, can just go straight through the membrane very simply. Carrier and channel mediated facilitated diffusion is when diffusion still occurs, but a molecule might be charged or large or an ion, something that needs help getting through the very lipid or fat-like phospholipid bilayer. So carrier and channel mediated facilitated diffusion is when a molecule needs help or facilitation to get through the membrane. And both simple diffusion and facilitated diffusion is the movement of solute. An example of a solute would be salt when it's dissolved in water. Osmosis is the movement of a solvent. It's the movement of water specifically. We won't talk too much about filtration. It happens a lot in the blood vessels, especially really small blood vessels called capillaries. They tend to push fluid out at one end and pull it back in, and that's a type of filtration that also occurs passively. So here we can see simple diffusion. Notice that over time, we have molecules that are bumping into each other, moving from the extracellular fluid space through the membrane into the cytoplasm. So it's a passive transport process because something is moving from high to low concentration with no energy being expended. This is also simple diffusion because there's no channel or carrier protein helping out these molecules. So they're probably small, they're probably lipid-like, they're probably not charged, they're definitely probably not polar. And the amount of concentration on one side compared to the other, how big the molecules are that are diffusing across the membrane, temperature, and solvent density can all play a role in how quick simple diffusion occurs. Simple diffusion, again, is when something moves straight through the membrane with no help from high to low concentration, and no ATP or no energy will be expended. Anything lipid-like can go straight through the membrane. If we were to use channels or carriers, proteins that are going to help move that across the membrane, it would be facilitated diffusion. You might see terminology like diffuses down its concentration gradient when we're talking about a passive process. A concentration gradient is just the difference in the concentration of like molecules in two areas, like on one side of a plasma membrane compared to the other side. Again, facilitated diffusion is going to use a carrier or channel integral protein. Facilitated means we need help to move something that's maybe hydrophobic, something like glucose, amino acids, something charged or large across the plasma membrane. This isn't as simple because these molecules are bigger, or they have something that doesn't allow them to move through the lipid or fat-like phospholipid bilayer. When you're looking at facilitated transport, we're still not using energy. We're still moving solute, and the solute is still going from a high to low concentration with no expense of energy. Oftentimes, we're going to be transporting materials as they attach themselves to either a carrier or channel protein, and carrier proteins tend to always be open. As long as a molecule fits, it can bind, and that carrier protein will change shape. Whereas a channel protein also usually always open, but we don't have to have any shape change when it comes to a channel protein. As long as the molecule fits, it can go through. So you can see in this image of facilitated diffusion, we're still moving solute or something dissolved in a solvent through the plasma membrane. But we're doing that by using a channel or carrier protein. In the images shown, the channel protein is on the left, and again, as long as the molecule fits, it can go through the channel. Whereas for a carrier protein, even though they're generally always open in one direction, and they must be specific enough to bind to a particular solute, and once they do so, they change shape. But this is still without any ATP expended. A lot of water, different ions, and monomers like glucose, sucrose, and fructose, or even amino acids like to use facilitated diffusion. So here we can compare the two. Notice on the left, we have simple diffusion. There's no integral carrier or channel protein needed for lipid-like or small molecules to move straight through the plasma membrane from high to low concentration. On the right, we have facilitated diffusion. We still have movement high to low concentration with no ATP expended, but this time we need help from an integral protein, either a channel or carrier. So the facilitation is the help needed to move a solute particle. We're also going to mention osmosis. Osmosis is also passive. Osmosis does not require ATP then, but it's the diffusion of water or a solvent as opposed to a solute. Simple diffusion and facilitated diffusion we're looking at the movement of a solute. Now we're looking at the movement of water. And if we're talking about the movement of water, it also moves from an area where there is more water to an area where there is less water. And if we are looking at a question, and we'll see a couple of examples, that tends to mean that water always moves to the area with more solute. And we can talk about osmosis and how it affects cells by looking at tonicity. Tonicity is the amount of solute in a solution, and we can use three terms to help us measure tonicity. And the measurement of tonicity is osmolarity, and the three terms of osmolarity are hypotonic, hypertonic, and isotonic. If a substance is hypotonic, it has to have less solute and more water than another solution. If a solution is hypertonic, it has to have more solute and less water than another solution. And substances that are isotonic have the same amount of solute in water as another substance. So we would only use the terms hypotonic, hypertonic, and isotonic when we're comparing two different solutions. And solutions are generally made up of solute and solvent, or solute that's dissolved in solvent. So an example of a solute would be salt, and it would be dissolved in water, the solvent. Here's another video that you can watch. Make sure to check out, again, this PowerPoint on Teams. So let's look more at osmosis. Notice that we have the water molecules outside the cell, and they can move through a channel protein to get inside the cell. But water is still moving without the use of energy through the plasma membrane from high to low concentration. So the main difference from osmosis compared to simple diffusion and facilitated diffusion is what we're moving. With simple diffusion and facilitated diffusion, we are moving a solute, a particle dissolved in something else. With osmosis, we are moving water, but all of them move from high to low concentration without the use of energy. That's what makes them all passive processes. So here we can look at a semi-permeable membrane. Something that's semi-permeable means some stuff can get through and other things cannot. This membrane would only let some stuff through while others can't make it through those pores. In osmosis, water always moves from an area of higher concentration of water to one of a lower concentration of water. And so if in a system like this, the solute, the dots dissolved in the water, can't move, the water will. And notice that the water is moving from left to right, because if there's less solute on that left side, that means there must be more water. And if there's more solute on the right side, that means there must be less water. So to reach equilibrium, or to have two solutions that, even though we have less of one and more of the other, that they both have the same concentration, the water is moving from high to low concentration. Or an easier way to look at it here, it's moving towards the area with more solute or more dots. So here's those terms again we've been talking about: hypotonic, hypertonic, and isotonic. And the definitions I gave you are listed out. When we're using these terms that are telling us about osmolarity, we have to be comparing two solutions, like solution A and B, or left and right, for the U-shaped test tube. The hypotonic solution has less solute than water compared to the solution on the right, whereas the hypertonic solution has more solute and less water compared to the solution on the left. And we're going to see that water moves from the left to the right through the semi-permeable membrane because the membrane is impermeable to solute, meaning the solute cannot move through the pores. But water can move, so it does move to the right to help equalize the concentration, because its goal is to produce an isotonic solution on both sides, where they both have an equal amount of solute and water concentration. And this can play a role in cells. We don't want cells to shrivel or swell up and bust, and eukaryotic animal cells don't have a cell wall to protect them. So if you place, for example, a red blood cell in a hypertonic solution, the water inside the cell will want to leave to go outside the cell where there's more solute. It'll make the cell shrivel or crenate. Isotonic solution is best for animal cells. It helps them maintain their regular shape, and for a red blood cell, it's this biconcave shape. Whereas if you put a red blood cell in a hypotonic solution, there would be more water outside the cell than inside, and that water would rush in. The water rushing in would cause a eukaryotic animal cell to swell and bust, or lyse. And cytolysis is the rupturing of a cell due to excess internal pressure, whereas crenation was the shriveling of a cell due to a lack of water. So osmotic pressure can change the shape of eukaryotic animal cells, so we want to be careful what type of solution we are putting our cells into. So here's just other images. You can see in the middle that we have a crenating cell, and then a swollen cell on the far right. If the cell that is in a hypotonic solution were to take in more water, it would likely bust. In a plant cell, we don't necessarily see lysis of the cells, but we can see plasmolysis, or the collapse of a walled cell's cytoplasm due to a lack of water. So plant cells don't crenate. Instead, their cell membranes pull away from their cell wall. And in a plant cell, if it's placed in a hypotonic solution where it starts to swell, that actually helps the plant. It makes it turgid. As long as it's not a waterlogged plant, it doesn't really result in a whole lot of negative for a cell with a protective cell wall like eukaryotic plant cells. So let's try some practice questions. We'll start down at the bottom. We're looking at the left and right situation, which are almost the same. On the left, we have a semi-permeable membrane. It's impermeable to solute, whereas on the right, we have a membrane permeable to solute and water. And we're going to ask ourselves the following questions for each scenario. 1, does solute move through the membrane? If so, which direction? 2, does water move through the membrane? If so, which direction? So let's look at the left situation. Let's see, does solute move through the membrane? Our answer is going to be no, because this membrane on the left is impermeable to solute. But water does move because the membrane is not impermeable to water. And water is going to move from the left to the right because it always moves towards the area with more solute. If we're looking at the situation on the right, water can move because this membrane is permeable to solute and water. And we're going to see that the solute moves from high to low concentration, whereas the water still moves from left to right or towards the area with more solute. Then if we move towards the top, we're looking at situations A, B, C, and D, and we're going to compare them to each other. A is blank to C. Look at A, it has more solute than C. So A is going to be hypertonic to C. All right, next is C is blank to B. All right, look at C and look at B. C has less solute than B. So C is hypotonic to B. Now look at B and now look at D. And B is blank to D. B has more solute than D, so B is also hypertonic to D. And now if we compare the last two, A and D, we find that they have the same amount of solute, which means they must have the same amount of water. So A and D are isotonic to each other, or you could say D is isotonic to A. All right, another clinical homeostatic imbalance that we can talk about is, again, using those terms isotonic, hypertonic, and hypotonic. You can clinically treat people with fluids. However, it is recommended never to give hypotonic solutions because that could result in dangerous lysing of red and white blood cells or busting of those cells. But we would usually use isotonic solutions and give those to patients if we just want to maintain a blood volume and pressure, especially if those need to be increased quickly. We could give a hypertonic solution if someone's become edematous, or if their tissues have been swollen, meaning fluid is moving out of their bloodstream and into tissues. And if we want to pull that fluid back into the blood, back into the bloodstream, we could administer a hypertonic solution into their vascular system. So we can see a clinical application for delivering certain types of intravenous solutions with different osmolalities. All right, so let's do a quick review. Osmosis is a passive transport process. Osmosis is looking at the movement of water, not a solute. Water is a solvent. If we put a cell in a hypotonic solution, it's likely to swell. If we put it in an isotonic solution, even though there's still going to be movement of water, it's an equal intake and export of water, so we're not going to see shrinking or swelling of that cell. And with a hypertonic solution, if we put a cell in that type of situation, it's likely to lose water and crenate or shrink. So here is a summary of passive processes. So make sure you go back and review simple diffusion and facilitated diffusion, which are both movements of solute, and osmosis, which is the movement of water. They all move from high to low concentration without the input of ATP or cellular energy. And next, let's look at active transport. You need ATP for active transport. It requires it. And we're going to see the use usually of some type of protein or a pump to help us move something from low to high concentration. And generally what we're moving is a solute. Anything that's very large, that can't pass through a channel, that needs help moving, again, from low to high concentration instead of down its concentration gradient, will require ATP. A lot of times we use the electrochemical gradient in order to power things like muscle cells and nerve cells. In order to restore certain parts of this gradient, we have to use active transport to move ions like sodium and potassium. And primary active transport uses a pump called the sodium-potassium pump because that's what it's moving. It's moving sodium and potassium across the membrane in separate directions. It actually generally moves sodium out of the cell and potassium into the cell, and it does this by binding to ATP and breaking it down and using that release of energy to change the shape of this protein pump. So primary active transport is when we directly use ATP to change the shape of a carrier protein, and this particular carrier protein is being named after what it moves, which is sodium and potassium. So here we can see that primary active transport. So we're noticing that sodium is the yellow dot, potassium is the green dot. You can see ATP in the kind of red star pattern. ADP and phosphate, when they come together, that creates ATP. So sodium binds and moves out, potassium binds and moves in. Notice sodium binds when ATP binds to the proton pump, and potassium binds and is able to move in when the last part of ATP detaches. There is also a secondary active transport, and this is a type of transport that just doesn't fit well anywhere else. But it actually has to be coupled to primary active transport because it doesn't directly require ATP. It kind of piggybacks off of primary active transport. We also have some definitions here, a symport versus an antiport system. What we just saw with primary active transport was an antiport system. We move substances in opposite directions. A symport system would move multiple substances in the same direction. Again, secondary active transport is going to indirectly use ATP, not directly. It actually relies on the ionic gradients created by primary active transport. And primary active transport on the left, you can see our example, does use ATP, and that creates a sodium gradient outside the cell that allows glucose to piggyback through a channel or carrier protein, depending on the situation, and move itself from low to high concentration. It doesn't happen a lot in the cell, as sodium here is diffusing back across the membrane for the situation on the right. It's moving as a co-transporter or with glucose, and it's driving that glucose against its concentration gradient into the cell. So the reason we're considering this active transport is because something's traveling from low to high, less so because it's directly using ATP. So make sure that you still kind of have this idea of primary active transport and other types we're about to look at as requiring ATP, because that's the most common situation. But we also do see the movement of low to high and count that as active transport. One of the last parts of active transport we're going to look at are different types of vesicular transport. Vesicular transport is active transport, and it's essentially using cell bubbles to move stuff in or out of the cell. Endocytosis is the process of moving particles into the cell by means of vesicles, and those vesicles form from the cell membrane. There's 3 types of endocytosis, and they are all active transport. And since they're endocytosis, they all are a part of bringing stuff into the cell. Think of penocytosis as cell drinking. It's the cell taking in solute dissolved in a solvent. It's taking in extracellular fluid. Phagocytosis, think of this as cell eating. It's when a cell takes in solid particles, and it takes in these solid particles or engulfs them. And then receptor-mediated endocytosis is essentially specific phagocytosis. It's when endocytosis occurs, but it's the cell using a receptor to go after a specific molecule it wants to take in. So there's a targeted solute particle with receptor-mediated endocytosis. So here are some examples. We have a macrophage, a real macrophage on the left, and it's eating bacteria. So it's performing phagocytosis, And that phagocytosis is to eat the bacteria and digest it. You can see kind of the cartoon of that on the right. Here also are some images showing you phagocytosis, pinocytosis, and receptor-mediated endocytosis. So I've already defined them, but take a look at receptor-mediated endocytosis at the top right, and notice that we're looking for a particular target molecule that will bind to receptors before something moves into the cell, and it moves into the cell by kind of collapsing in on itself and creating a cell bubble, and the cell bubble is what we consider a vesicle. And then exocytosis is also vesicular transport. This is vesicular transport wherein we are moving something out of the cell. Something like mucus or neurotransmitter or hormones could be gotten rid of or leave the cell via exocytosis. But we also get rid of waste products via exocytosis from cells. So here we're seeing a vesicle migrate to the plasma membrane, bind and release contents. And this can be waste products, this can be other stuff that cells make, but exocytosis is always when things are ejected from the cell. And both endocytosis and its 3 types, as well as exocytosis, are all types of vesicular transport, and all vesicular transport is active transport. So here is a summary of our active processes. So primary and secondary active transport, exocytosis, phagocytosis, pinocytosis, and receptor-mediated endocytosis. So those last 3 are all endocytosis, and everything requires ATP, even indirectly if we go back to secondary active transport. And that is it for this PowerPoint.